Good morning, students. Today, very important topic: health and diseases. AIDS. AIDS means what is the stands for the AIDS? AIDS stands for acquired immunodeficiency syndrome. Acquired. So acquired means it is a developed after birth. It is not a congenital disease. It is acquired during lifetime. Any infection of HIV virus, it causes the AIDS. Acquired means it occurs in the during lifetime. It is not a birth developed disease. So it is not a congenital disease. Genetic disorders are the congenital disease. Immunodeficiency after infection of HIV virus, they infect to immune cells. Target for the immune cells. Progressively, their number is decreases. The persons causing more infections. Lack of immunity power means immunodeficiency. So, cause the immunodeficiency. After immune deficiency causes syndrome. Syndrome means group of disorders. Group of symptoms appear in the AIDS person. So AIDS mainly caused by infection of HIV virus. HIV stands for human immunodeficiency virus. That virus target to the immune cells and it decrease the human immune system. It causes many symptoms are appear. That final condition is AIDS. First time AIDS virus, HIV virus identified in the United States of America in the year 1981. In India region identified 1986. So onwards, many persons are die due to infection of HIV virus. So many persons are die. More than 25 millions are peoples are die due to infection of HIV virus. Last 25 years. So HIV virus, so it is causing not curable disease. Not curable disease is AIDS. So only contain the partial treatment is available. The partial treatment, once a virus is infected, it is not curable. So finally leads to death. Depends on person immunity power. So persons are the once infected, leads to death. Only available of partial treatment, not completely curable disease. AIDS. How to infection of HIV virus. Next, infection of HIV virus. How to infection HIV virus. So mostly it infected through sexual contact. Any person is HIV virus infected, the sex with non-infected persons automatically easily transfer to the other persons. So infected persons are spreading the non-infected persons through sexual contact between life partners, between unknown partners, or multiple partners. So one person contact with multiple persons, they easily spreading to HIV virus to all persons. So first infection is through sexual contact. Next, second one, through blood transfusion. So every person before the blood donate screening the HIV virus. So it is a screening negative. So after can cause the donation of blood. So without screening of HIV, negative or positive. So there is spreading by blood transfusion. Blood transfusion easily spreading to others. Next one, third one, transmission of HIV virus mostly by infected syringes and needles. So HIV is mostly spreading through the sexual contact and blood transfusion. Infected needles. Private, public clinics only using for the disposal syringes, needles. So that is important, prevent the spreading of HIV virus. Sometimes one person using needles or syringes inject to the many persons. If anyone is infected, automatically the infected persons needles spreading to all. So mostly also HIV transmission through infected needles and syringes. Another transmission between mother and fetus. One of the child connected between the mother through the placenta. So placenta. So placenta is sometimes not filtrate the HIV virus. So placenta is one of the barrier spreading of the disease. So sometimes spreading to the babies or fetus. So HIV also transmission between mother and fetus through placenta. Through placenta. So mostly many persons are spreading by this four process. First one is HIV is a STD. STD means sexually transmitted diseases. So transmission through sexual contact. HIV virus living in the body fluids, mostly in the blood. They are transferred through the blood transmission. Next, finally, through placenta. Sometimes, so spreading to the babies. Some of the time not spreading to the babies by using of the mother using retroviral drugs. Retroviral drugs control the spreading of disease between mother to fetus. This is the most four steps are the infection of HIV to others. Next, structure of HIV virus. HIV your human immunodeficiency virus. So HIV virus, it contains the genetic material. What is the genetic material? Single stranded RNA material. So virus contain the single stranded RNA material. Two single stranded RNA materials inside contain the enzyme, reverse transcriptase enzymes. Next, surround genetic material HIV surrounded by protein coat. Next, outermost contain the double layered lipid membrane with attaching to glycoprotein molecules. Their attachment with glycoprotein molecules. Glycoproteins are the two types of molecules or preparation. Carbohydrate and proteins. They contain the two types of units. Small units. Glycoprotein 41. Large unit. Glycoprotein 120. Very important glycoprotein molecules attaching to host cell membrane. So the protein molecule, glycoprotein molecules 120 attaching to the host cell membranes. HIV virus infect to the infect to the host cell. Only HIV virus infect to the immune cells only, not infect to the all cells. Infection only immune cells, the host cells. The host cells contain the surface cell contain the one markers. Surface contain markers. CD4 markers. So what is the cells are infected by HIV virus? The infective cells, immune cells, which contains CD4 markers. The target for macrophages and infected to the immune cells T helper cell TH cell. So CD4 markers present on CD4 marker present on the B lymphocytes and dendritic cells So HIV virus only target or infected to attaching to and enter into the CD4 marker cells. The target mostly macrophages and T helper cells. Only some cells cause the destroyed by the HIV virus. After entering, produce multiple copies of HIV virus. After then, destroy the host cell. Some of the cells are the not destroyed by the HIV virus. Certain cells are acting as a HIV factories. Produce the multiple copies of HIV virus. Next, how to produce a HIV virus in host cell. It is a clearly understanding about the observation of with the help of diagram. This is a HIV virus. Target for the CD4 markers. HIV virus. Attached the CD4 markers with help of glycoproteins 120s. After attachment, only release viral RNAs remaining present outside the host cell. That host cell outside is called as a ghost. That RNA enter into the host cell cytoplasm. in cytoplasm along with contain the reverse transcriptase enzymes. The two RNA strands with help of enzyme. What is enzyme name? Enzyme name is the enzyme name reverse transcriptase enzyme. Convert the viral RNA into viral DNA. That viral DNA is complementary DNA. cDNA, complementary DNAs. cDNA with help of enzyme reverse transcriptase enzyme. The process is reverse transcriptation. You know the transcription? Transcription means formation of RNA from DNA with help of enzymes. The process is transcription. Next, RNA collect the amino acids, prepare protein. The process is translation. The general process of transcription. But HIV virus, retrovirus, which already contain the RNA material. That RNA converted into DNA. That formation is reverse transcription. Formation of DNA is reverse transcription with the help of enzyme reverse transcriptase. So from the viral DNA, that viral DNA enter into the nucleus, bind to host cell genome, bind to the one of the chromosome, DNAs incorporated of host genome with the help of enzyme. Enzyme is integrase. HIV virus also contain the enzymes integrase. What is the importance of integrase? So viral DNA bind to host DNA. That process is integration. After binding host cell DNA, prepare the DNA prepare the translate RNA materials This is a viral RNAs. So, host cell nucleus produce new viral RNAs. They produce the multiple copies. So RNA again produce the translate protein coat. That protein coat surrounding and prepare the viral parts. So all these assembled in the host cell. Now host cell prepare the multiple copies of virus. Host cells are the bud of production of the new virus. After production, cell will be die. New virus release into blood circulation. Again infect to new host cells progressively decreases day by days, decreases the numbering of the immune cells. So persons, any HIV virus infected persons cause the immunodeficiency. So it is new virus target ready to infect to the other cells. So that is a process formation of new virus in the host cell after infection. The symptoms are appear. So after infection of HIV virus, not immediately cause any symptoms. HIV virus contain the long incubation period. Incubation period is take the up to 5 years, not show any symptoms. Depends on the immunity power of the person. The first symptoms appear period is after incubation period. So early infection, early time first it cause a bouts of fever, not a continuous fever. So it cause a fever. Next early infection it cause a symptoms diarrheas, loose motions, and cause the most observed character of HIV infection, weight loss. Next, loss of appetite, not interested for the taking of food, appetite. These are early symptoms. Progressively, the viral load increases. Damage to more immune cells, especially macrophages, T helper cells. Some of these cells are not destroyed. They are living. Only target for the some cells. So gradually, within the 5 to 10 years, develop the AIDS, acquired immunodeficiency syndrome. That time easily infection of the many parasites, infection of HIV virus, Mycobacterium, tuberculosis, cause the TB, develop the TB, and next HIV infected persons develop the infect the immunity part, infect the lymph nodes, most of the lymphatic tissue present in the respiratory part, intestinal part, lack of security. So immediately it causes typhoid, next pneumonia, other infections also easily infections occurs in the HIV persons. Next, lack of immunity power. HIV infected persons. One of the virus also easily infected. Toxoplasma. So it will easily cause a other syndrome characteristics. So HIV infected persons lack of immune power. Immune cells are give the protection to the cancers. So lack of protection, the persons are easily causing cancers. One of the type of cancer, Kaposi sarcoma. One type of skin cancer. So HIV persons, T helper cells. You know the T helper cells. They involved in the two types of immunities. One is humoral immunities and T helper cell involved in the cell mediated immunity. So decisively affect the two types of immunities. Next to diagnosis. Symptoms after diagnosis. Detection of the HIV virus. So HIV virus against produce antibodies. Detection of antibodies. The test is ELISA test. Enzyme linked immunosorbent assay test. One of the detecting of HIV antibodies against HIV virus. HIV virus, this test not detect the HIV virus. Only detection of the antibodies. So it is a early diagnosis test is ELISA test. After diagnosis, sometimes also negative also show the positive. Other antibodies are detected, activating of other immune cells. That's why after then HIV virus conduct the another test. After ELISA conducting test is confirmation test. Confirmation test is Western blot test. Western blot technique. Western blot technique detect the HIV virus protein coat. So under the process of gel electrophoresis. So detection of DNA is any DNA material detection. In the process of DNA fingerprinting, detection of DNA fragments is DNA detection is Southern blotting technique. Here the Western blot detection of protein coat. Two tests are available for the detection of HIV virus. Treatment. Especially treatment is using retroviral drugs. HIV virus not completely curable. Only increase the HIV infected person's lifespan by using antiviral drugs, antiretroviral drugs. These drugs delay the replication. Here they inhibit the one of the enzyme is inhibited. Reverse transcriptation. Reverse transcriptase enzymes are inhibited by antiretroviral drugs. Delay the replication. So decrease the increasing of viral load. There are two. One of the drug name Jido, Udin, or scientific name azithromycin. Is a drug also used in the pregnant women, delay the reverse transcriptation and delay the and prevent the spreading of disease from mother to infected mother to fetus. Next, prevention of HIV virus. Preventive methods. Next, preventive methods. Preventive measures. First of all, awareness, creating awareness about the all society or peoples are create the awareness, awareing about the information of HIV and AIDS. So HIV or AIDS, once infected, it is not curable disease. Only some partial controlling measures, partial treatment is available. Only increase the lifespan. So HIV is killer disease. How to prevent spreading of disease? Preventive methods. Prevention spreading of disease. First, awareness about the sexual contact. So HIV is a STD. To avoid the sexual contact with multiple partners, unknown partners, is the first prevent the spreading of disease. Next, using disposal syringes and needles in the private public clinical centers. Next, third one, blood is collected from the blood banks. Blood banks definitely screening of the HIV virus prevent the spreading of disease. So next, HIV also infected through the TD abuse. The persons are avoid the TD abuse, especially drugs are injected through the intercourse of the veins, infected needles and syringes. Avoid the TD abuse. Lot of awareness created by the NACO, National AIDS Control Organization. Lot of awareness created entire country wise. Next, other awareness created by the NGOs, non-governmental organizations, Red Cross, Lions Club, anyone is create the awareness. One of the WHO also create the awareness. World Health Organization. So every December One is announce the World AIDS Day. The day they create the awareness about the AIDS. So lot of awareness created by the many organizations, centers spreading throughout the world. Create the total awareness. It prevent the spreading of disease.